So with that, I'd like to introduce the speaker of today's webinar, Dr. Ricardo Salvador from Neuroelectrics. Ricardo is an expert in non-invasive brain stimulation. He's leading the modeling team at Neuroelectrics. He published more than 80 papers in the area of modeling and brain stimulation. His experience includes international projects and collaboration with several teams worldwide. And his main research interests focus on the development of multifocal transcranial electrical stimulation. So Ricardo, the micro and screen is yours.
Okay, thank you, Rafael, and hello to everyone, and welcome to this webinar. The title of this webinar is a little bit ambitious, everything you always wanted to know about tES. So as you might imagine, we have a lot of ground to cover, and I won't have time to go into much detail about every specific topic that we'll cover in this webinar. However, I think this is a good entry point. I mean, I hope this is a good entry point for you to learn more about any specific topics. And in the end, if you have any doubts about something that I didn't mention, please just let us know. So in this presentation, we will start with an overview of the historical background and a brief introduction to this technique. Then I will mention aspects related to instrumentation and setup of this technique. I will discuss the mechanisms of action of tES. I will then discuss the importance of computational head models in dose parameters planning, and I will address some of the most common practical applications of this technique. So to start, I would like to go over a brief historical background of this technique. I like to do this usually because, at least for me, it's not clear at all how this technique evolved and how people started thinking of using electrical stimuli for health benefits. So apparently the first reported use of electrical stimuli for clinical applications comes from the Egyptian times, where they reported the use of the torpedo fish, which is an electrical fish, to cure certain diseases like migraine or muscle pains. And the torpedo fish, of course, is an electrical fish, so it's a fish that uses electrical discharges to hunt for prey and for self-defense. And they would use the electrical discharges it produces to help patients with these problems. Then the Romans described this process as well. And many years later, Luigi Galvani did a very famous experiment today. He had in his experimental table dissected frog legs and accidentally touched a metal conductor that was charged electrically. He touched the frog legs with that metal conductor, and the frog legs suffered a contraction. And Luigi Galvani saw that there was then a connection between electrical stimuli and biological systems. Thus, he is considered the father of bioelectricity. In 1862, Guillaume Duchenne used electrodes to stimulate the facial muscles of patients non-invasively, although I can imagine this was quite painful for the participants. And in 1951, Wilder Penfield used, of course, invasive electrical stimulation to map motor function in the primary motor cortex and also in the somatosensory cortex. But it was only in 1998 and 2000, with the works of Alberto Priori and Michael Nietzsche, that the first usage of transcranial electrical stimulation was done. And this is the technique that we'll discuss today. So non-invasive electrical stimulation. Electrical stimulation is part of a group of techniques that can be framed within bioelectromagnetic phenomena. And these phenomena, they happen because in the tissues, in the intra and extracellular medium, we have ions, and these ions have electrical charges. So our tissues in the head, in particular, they can conduct electrical signals. And in particular, in the head, in the brain, the neurons and glial cells, they have proteins in their membrane. And these proteins, they can regulate the flow of ions across the membrane. And by doing so, they can generate and regulate the membrane voltage difference between the inner and the extracellular medium, and that's called the transmembrane potential of the cells. And they can generate different type of responses. They can generate passive responses, which are similar to the charge and the discharge of an RC circuit, so a circuit with a resistor and a capacitor. They can generate active responses. These are also known as action potentials, so nerve impulses that can propagate along a neuron without attenuation. And of course, when the action potentials reach the end of an axon, they will elicit the release of neurotransmitters that will then influence the membrane potential of another cell, and those changes in the membrane potential of the postsynaptic cell, they're called postsynaptic potentials, which are all, also a form of passive responses. So this electrical nature of the brain allows us to read the electrical signals non-invasively. So with electrodes placed outside, we can of course read electrical information from the brain. In that technique, you all know it as EEG, electroencephalography. And of course, in transcranial electrical stimulation, we can use electrical stimuli that are applied non-invasively. So with electrodes placed on the scalp, we can use those electrodes to generate an electric field that will influence the membrane potential of the neurons. Now, I will explain exactly how this electric field will influence the state of the neurons in the next couple of slides. But before we do that, I would like to go over the instrumentation and the setup for this technique. So basically, you will need the following components. You will need an electrical stimulator. The electrical stimulator connects to electrodes placed on the scalp via cable connectors. And the electrodes are placed in standard positions of the head. The standard positions are the same as those of the 1010 or the 1020 EEG systems. And they are placed in those positions with head caps. These head caps guarantee that from session to session, the positioning of the electrodes is maintained approximately constant. Stimulators are current control devices, which means that you specify a current and the device maintains and controls that current during the stimulation. The device can generate currents from 1 to 4 milliamps. So the maximum current per channel is around 2 milliamps, and the total injected current, so the sum of all the currents going into the brain, can go up to 4 milliamps. Modern devices, they support multiple channels. For instance, the device that we have here in this picture, Starstream 32, can support up to 32 channels. In theory, to perform TDCS, you only need, or tES, you only need 2 channels at most, because you need the stimulator, the cables, 2 electrodes, and the head to close the circuit and allow for the current to flow. However, you can have more than 2 channels. You can have, in this case, up to 32 channels. And the idea here, the only constraint here is that the sum of all the currents on all the electrodes needs to be 0 at all times. The waveforms, the temporal waveforms of the currents in the electrodes can change. And this technique is called transcranial electrical stimulation, but depending on the waveform of the currents, you can have different names for the different ways of applying this technique. A very common name is TDCS, transcranial direct current stimulation, in which the current is maintained constant throughout the application. And of course that anodes are the electrodes that inject current into the brain, and cathodes are the electrodes that extract the current from the brain. So current is positive in the node in the anodes, and it's negative in the cathodes. TACS, transcranial alternate current stimulation, is a form of tES in which the current varies sinusoidally with time, and the frequency of variation is usually the low frequencies of EEG, so from DC to a couple of hundred of hertz. And finally, TRNS, transcranial random noise stimulation, is a form of tES in which the current in the electrodes varies randomly in time with the uniform frequency distribution, so from 0 to 600 and something hertz. These waveforms, these currents, they're applied for 10 to 40 minutes, depending on the protocol, sometimes slightly more, sometimes slightly less. Regarding the electrodes, the first generation of electrodes, so the first electrodes that people started using in this technique, were large saline soap sponges. The areas were between 25 and 30 to 35 square centimeters. Saline solution was necessary because obviously the sponge, without being soaked in a conductive medium, couldn't allow for the conduction of the current. However, as multi-channel devices became more and more prevalent, the size of the electrodes needed to decrease, obviously, because we can't fit, for instance, 32, 35 square centimeter electrodes in the head. So the electrodes that you see here in this picture are actually very small electrodes, 1 centimeter radius, and they are metallic electrodes. They're made of silver, silver chloride electrodes, and they have a conductive gel located underneath. Conductive gel, so it's just a conductive material that is really important to make sure that stimulation can happen, and I will describe the reason for that in a future slide. So this picture here shows the different types of electrodes. You have the sponge electrodes that need to be soaked in saline solution, and you have the smaller PyStim electrodes that need to have the gel underneath. This is the process of soaking the electrodes in saline solution, and this is the process of injecting the conductive gel underneath the electrodes. So regarding the mechanisms of action of this technique, we need to start by understanding what happens when we turn on the device. So imagine this case. Imagine that you have the electrical stimulator that's connected to a lot of electrodes in the scalp via the different cable connectors. What happens when we turn on the device? The first thing you need to understand is that the charge carriers in the wires are electrons, because the wires have a metal connector. However, the charge carriers in the head, they're ions, like I mentioned in the beginning. So in the interface between the electrodes and the head, there needs to be a chemical reaction that converts from one type of charge carrier to another type of charge carrier. And this is where the gel plays a big role. If the gel wasn't present, the voltage difference to generate this chemical reaction would be much higher than the values that our current devices can generate. So in order to enable this, we have a conductive gel that lowers the impedance of this interface between the electrode and the head, and allows for this reaction to happen at much lower voltage differences. If the gel isn't enough, or if there isn't a good contact between the gel and the scalp and the electrodes, you will get a warning that the impedance is too high and the stimulation simply won't start. After you got the current flowing, we physicists, we like to think of current with another concept, which is the concept of electric field. Electric field in physics is just a vector. It means that if you have an electrical charge and that electrical charge is in the presence of an electric field, that electrical charge will feel a force along the direction of that electric field that will be proportional to the strength of the electric field. Now, if you have a bunch of charges and you have them in a region where an electric field exists, those charges will move, thus establishing a current. So currents and electric fields are similar concepts in conductive media, such as the brain. And we like to think of the electric field distribution in the brain when we generate, when you turn on the device. And it doesn't matter in how we calculate this electric field distribution. This will be discussed later, but What you need to understand about the electric field is that in each point of the head, you'll have one direction, which is the direction of the electric field, and a specific strength for the electric field in that region. Okay? Now the question is, how does this electric field help us understand the effects that this technique has at the level of the neurons? So the basic concept here is that the electric field will establish a force in the ions, in the extracellular and intracellular media, and that will lead to changes in the transmembrane potential of neurons. Okay? And the mathematical theory for describing how this electric field interacts with the neurons is a relatively complex theory, but basically what you need to understand is that the component of the electric field, the direction of the electric field, that will predominantly affect the transmembrane potential of neurons is the component of the electric field aligned with the main direction of the neuron. And what neurons are we talking about in the brain? Well, the neurons that we think are more susceptible to tES are the big pyramidal cells that exist in the cortex. And these big pyramidal cells, that they tend to align themselves perpendicularly to the cortical surface. So as one would expect, the component of the electric field that's more likely to affect the membrane potential of these cells is the normal component of the electric field. And how does this work? Well, if the normal component of the electric field is pointing into the cortical surface, it's pointing into the soma of these pyramidal cells, this will lead to a depolarization, so an increase in the transmembrane potential of the soma, which will make this cell more easily excitable. So it's increasing cortical excitability. On the other hand, if the electric field is pointing out of the cortical surface, then this would lead to an hyperpolarization of the soma. So it will make these cells less excitable. This also forms the basics for that simple theory that under anodes, there's an increase in cortical excitability, because under anodes, the field is predominantly going into the cortical surface, whereas under cathodes, the electric field is coming mostly out of the cortical surface. As you can see in this picture, this isn't as clear-cut as that, though. For instance, in this picture, we're roughly in the region of the head that's under an anode. And you'll see that here, in fact, the electric field is coming into the cortical surface. But for instance, here, the electric field is coming out of the cortical surface. So in order to understand what's going on, you often have to consider large populations of neurons and not just really focalized regions of where the electric field might interact with the neurons. Another potential mechanism for action is through other components of the electric field. For instance, the tangential component of the electric field will affect components of the neurons that are aligned with this electric field. In this case, axon collaterals. And this is also described as one possible mechanism that might influence transmembrane potential. Now, the changes in membrane potential that are elicited with electric field, they're really small in tES. They're about 0.27 millivolts per volt per meter of electric field. Now, for you to have an idea, the electric field typically induced in tES is around 1 volt per meter at the most. So we're talking here about a polarization that's really, really small, 0.27 millivolts. It's way smaller than the threshold for generating action potentials. So we're generating sub-threshold responses from the neurons in this technique. So the question here is, what can we do with such small changes in membrane potential? Why would this have an effect? Well, there are studies that show that even these small changes in the membrane potential, they can affect the timing of generation of action potentials. So the cells in the brain, they aren't relaxing all the time. They're having action potentials, they're communicating with one another. And as they have the action potentials, if the cell is slightly depolarized, so if the excitability increases slightly, the timing of the action potentials is reduced. So the frequency of action potential increases. Whereas if the cell is hyperpolarized, the timing of the potentials, it actually decreases. So the frequency of the action potentials will decrease. And these changes in action potential timing might also lead to changes in cortical excitability. And also, this also provides a way for the ACS to interact with ongoing cortical oscillations. So cortical networks, they often communicate with specific frequencies in the brain. And if you fine-tune the frequency of your TACS to match the frequency of that ongoing cortical oscillation, you might have a stronger effect of the technique, and you might influence that particular network. TRNS works with different mechanisms. It works with something called stochastic resonance, which is basically a mechanism by which, if you have a subthreshold signal and if you inject some noise to it, that signal becomes supra-threshold. And that also is to the fact that TRNS is mostly excitatory and cortical excitability, as shown in a number of studies. Of course, that these small changes in cortical excitability, they don't last long after the stimulation ends. However, if you repeat this stimulation across a period of several weeks and across several sessions, you'll actually induce long-lasting side effects that are due to ambient plasticity-related mechanisms. In this paper that came out in 2019, the authors induced long-term potentiation using a well-known stimulation protocol, theta burst stimulation. And they did this in two conditions. In one condition, they had an excitatory electric field being applied in the cell. And in the other one, they had an inhibitory electric field being applied in the cell. And what they found was that the responses to this long-term potentiation were boosted in the case of the excitatory electric field, and they were reduced in the case of the inhibitory electric field. So this provides the basis for the application of this technique in most protocols that are available today. So instead of applying just stimulation in one day, we usually apply it once every day during the week, and we repeat this for several weeks. And there are a number of studies that show that this type of protocols, they have long-lasting effects in a number of neuropsychiatric disorders, and we will discuss these applications later. However, many studies, they also point out that there's a considerable inter-subject variability in the response to these stimulation protocols. And inter-subject variability is something that is to be expected whenever biological systems are involved. We can see it right now in the COVID-19 crisis. Some people, they have more serious symptoms, whereas other people don't experience such severe symptoms. And this is related, in this case, this is related to genetic makeup, individual genetic makeup. But in the case of non-invasive brain stimulation protocols, this sort of variability results not only from genetic makeup, but also from factors that are more related to non-invasive brain stimulation, namely stimulation intensity, electrode sizes and shapes, and the position of the electrodes, the geometry of the electrodes. These parameters, they're what we call dose parameters in tES. So they're the controllable parameters that affect the electric field. And I think that from the previous slides, you now understand that knowing the electric field is essential for understanding the effects and predicting the effects that a given stimulation protocol will have in the patient. So if this electric field is so important, we've spent the last couple of slides talking about how important the electric field is. How can we measure it? It's a simple question, right? The problem is that there's no way to effectively measure in vivo the electric field. There are studies out there where researchers have done this. In this study, the researchers took advantage of subjects that had electrodes implanted for other reasons, and they used those electrodes to probe the electric field in these specific positions in the brain. And in this other study, the researchers created a very specific MRI sequence that is susceptible to the very small magnetic fields that are generated by the currents induced in the head during tES. The problem with these two techniques is that they generate only a very low resolution spatial map of the electric field. And we can't really use these maps to great effectiveness. So we need a high resolution map. Luckily for us, we can build such maps using computational head models. And in the next slides, I will explain the importance of these computational head models in predicting the dose parameters in an experiment in TCS. So this presentation isn't about computational head models. We will go over something related to computational head models, some concepts, but we will have a webinar in the future where we will introduce these topics into more detail. And I highly recommend that you check out that webinar if you're interested in this. Computational head models in tES, they can be created using different methods, but all of those methods, they follow essentially the same pipeline. So the input to create a computational head model is usually a structural MRI of the brain. And that structural MRI of the brain is very important, and MRIs of the brain are usually a good candidate for that structural information that we need to provide, because they have very good spatial resolution. They have very good contrast for the types of tissues, for the soft tissues that we're interested in here, and they don't use ionizing radiation, unlike CT, which employs ionizing radiation. So after you've got your MRI, you need to segment it, so classify each voxel into each specific type of tissue that you want to include in your head model. And there's, again, a number of algorithms you can use for that. After that, you get the masks for each tissue, and you can create a 3D model of those masks. This 3D model is very detailed in terms of geometry, so it captures the geometry of a subject. And in this stage, you can also include in the head model representations of the electrodes that you'll use in that experiment. In this case, we're modeling the same type of 1 centimeter radius electrodes that I mentioned before. Once you have your 3D head model, you will want to calculate the electric field in that head model. And to do that, it's known from physics that you need to solve an equation that's called Laplace's equation. And that equation looks fairly simple, but it's very hard to solve that equation for a complex geometry like the head. So what we need to do is to use computational methods to solve, numerical methods to solve Laplace's equation. And usually in these studies, people use something called the finite element method, which is used in many applications and in particular in this one. To solve that equation, you'll need to provide some parameters to the model, namely, of course, the currents in the electrodes, and also the conductivity, the electrical conductivity of each of the tissues. The electrical conductivity is basically a parameter that tells you how well a given tissue can conduct electricity. And after you calculate the solution, you get a map of the electric field in the brain, for instance, and that map has a really high spatial resolution. The resolution is typically slightly lower than 1 millimeter, so it's a sub-scanner resolution. There's two things you can do with these models. One thing that you can do is, of course, look into montages that people typically use in their experiments, like the big sponges montages, and determine the electric field properties in those montages in a retrospective manner. This is important in order to shed some new light into the results of a previously done experiment, or to study the physical properties of the electric field in tES in general. But much more interesting, you can use these models to optimize the position and the currents of the electrodes to maximize the electric field in a particular target. So this is what we'll discuss how to do next, and this is what's called montage optimization in tES. And this forms the basis for dose parameter determination that I mentioned previously. So how can we do this montage optimization in a study in a practical manner? So the first thing you'll need to do is to build a head model using the methods that I discussed before. And this head model should include lesions if they're present in the subject, like craniotomies, for instance, because those lesions can have a very big impact on the electric field distribution. Once you have that, then you need to define a target for stimulation. Now, this target for stimulation usually comes from the clinical partners that are working with you in your study. For instance, the results that you see here are from a trial that we conducted with patients with focal epilepsy. And the information that we would get from the clinicians was the location of the focal epileptogenic hotspot. And after you determine the position in the MRI, we then map the position to the reconstruction of the brain that we made from the head model. And after you have the region, you need to decide what's the mechanism of interaction of the electric field with the neurons and what's the target electric field in that area. So in this study, we used a simple model that it is the normal component of the electric field that interacts with the neurons, and that if the normal component of the electric field is pointing into the brain, that will lead to an increase in cortical excitability. And if the electric field is pointing out of the cortical surface, that will lead to a decrease in cortical excitability. Since in the epilepsy trials, we wanted to decrease excitability in the focal region of the epilepsy, we would assign to that area a target electric field that would point out of the cortical surface. So an inhibitory electric field. And after we feed that information about the target and the target electric field in that area to an algorithm, to a numerical algorithm, we can extract the positions and the currents in the electrodes that will effectively minimize the difference between the electric field induced by the montage and the target electric field. Okay? I won't go into many details here about the numerical details of the optimization algorithm. Again, that will be the focus of a future webinar. One thing that you need to understand there, though, is that there are many constraints that go into an optimization. For instance, one thing you can constrain is the strength of the electric field. You can have an electric field that's relatively mild, or you can have a stronger target electric field. And increasing the target electric field to a higher value will increase the average electric field on the target, which might be a desirable thing. But if you apply that in a population of subjects, it also leads to a bigger variability in the results of the electric field. And that is because the optimizer won't have enough current budget to optimize for the electric field if you're being too demanding in the value of the electric field that you're asking for. Another thing that you can constrain, of course, is the total number of electrodes and the current that you inject in each of the electrodes. We have a presentation about the importance of these optimization parameters in these sorts of algorithms. This presentation was initially thought to be presented in the 7th International Conference of Non-Invasive Brain Stimulation. However, unfortunately, due to the current crisis, that conference was postponed. So we're still making the poster available by following this link. I highly recommend that you follow this link. You'll also find there a video, a presentation that explains this poster in more detail. The patients that we used in this study were from an European project that Neuroelectrics and Starlab are involved in, which is the STIPED project. The STIPED project is a very ambitious European project in which we're using personalized montage optimization to target patients that we have healthy controls, we have ADHD children, and we have ASD children and teenagers. And if you're interested, please visit the website for the STIPED project, and you'll find more information about the project there. My colleague Maria Chiara also has another very interesting poster about optimization, montage optimization, in this case in minimally conscious state patients. And these patients were part of a study, the another project, the Luminous project, in which Starlab is a partner. Again, I highly recommend that you follow this link and visit this project if you're interested to learn more about this. And if you follow this link, you'll find more information about the poster from my colleague. Now, the targets that I mentioned before, they're highly localized targets, but that's not always the case. It may be so that you have a network of targets that you want to reach in your stimulation protocol. In this work that we did together with Fisher and colleagues, we determined the regions in the brain that were correlated with the seed point located on the left motor cortex. So we used resting state fMRI to determine the regions that were correlated and anti-correlated to that seed point located on the left motor cortex. And then the rationale for stimulation was the following. The regions that were positively correlated with activity on this seed point, we marked for excitation, whereas regions that were inversely correlated to the seed point, we marked for inhibition. And after we applied montage optimization for that target map, and we tried the effects of that montage on motor evoked potential magnitude, we found that this approach led to a bigger increase on the motor evoked potential amplitude than the traditional approach where they used the anode over C3 and the cathode over the contralateral eyebrow. You can also use this approach to reach deeper sites with cortical stimulation. So a problem with tES is in reaching regions in the brain located very deeply. And the problem with that is that a lot of current that you inject in tES is actually shunted through the scalp. It doesn't really reach the brain. And so it's hard to reach deep areas. So one thing you can do is to get maps of the cortical areas, so the superficial areas, that are correlated with activity of that deep area, and then stimulate using the same sort of rationale that I described previously. And this is also described, this rationale is also described in this paper from 2018 that I highly recommend that you read when you have a chance. So talking about practical applications now. tES has a number of described practical applications. So during the years since the first experiments using TDCS, people realized that this technique had a potential big impact on a number of conditions, and they started publishing a lot of potential applications. However, of course, not all the studies are the same. Some studies are done with more patients, some studies are done with less patients, so you can't really extract the same conclusions from all the studies. So in 2017, Lefaucheur and colleagues, they did a review of all the papers that had potential applications for, in this case, TDCS. And they came out with a list of the applications for which there was stronger evidence for a possible effect of stimulation. And those applications were fibromyalgia, major depression, addiction, craving, and to a lesser extent, lower limb neuropathic pain. I'm also including here epilepsy because in a previous clinical trial that we did with Boston's Children's Hospital, we saw good results of the application of the same sort of rationale for optimization that I mentioned before. Now, you'll notice that next to these three applications, there is also a montage. And that montage is the montage that the authors of this paper identified as being the one that most likely is beneficial for the patients. Now, this goes against what I told before, because what I told before is that to get the montage parameters, you need to create a head model, you need to do montage optimization, you need to have target information. But here you just have a recipe, right? You have the positions of the electrodes, and you just apply this to all the patients. Now, we still believe that these are only first-order solutions, and that if you really want to improve the results and minimize variability between the effects of stimulation across subjects, then you need to use more complicated models. And in fact, we believe that not even just a model that includes that models the brain as passive, so a model like I described before, biophysical brain model, not even just this brain model includes all the relevant information. There's another layer that you want to add to the models of the brain, which includes the effects that the electric field has on networks in the brain, on networks of neurons. So a physiological brain model that uses neural mass models, for instance. And the idea is to combine these two layers into what we call an hybrid brain model. And then this hybrid brain model will be a much better way to do therapy design. So to optimize and to get the parameters, the dose parameters that will have an effect on a particular application. We believe this so much that we submitted a project, and we were granted a project by ERC to control epileptic brain networks using these hybrid brain models. This is a really interesting project. We call this the Galvani project. And if you follow this link, you'll find more information about this project. Incidentally, we're also in the period of hiring people for the project. So if you're interested and if you think you have the right skills for this project, please send us a job application. Another important parameter for the application of tES is, of course, related to safety and tolerability issues. And luckily for us, there's not much to report here. So tES is usually considered a safe technique. There's the possibility of the most commonly reported side effect is redness and a tingling sensation under the electrodes. This usually tends to disappear after stimulation. And there's also a possibility for generation of lesions in the brain for very high current values. This has been described in animal studies, but this isn't a real threat for humans because the threshold to induce such lesions in animals is 3 times higher than the electric fields that we're generating in humans. So we're working well within safety margins here. Another important concern is related to metal implants, since many subjects that we do stimulate, they have some sort of metal implant in the head. These metal implants, they are likely non-conductive at the low voltages that we generate in the brain with tES. So they actually function as isolators instead of conductors. However, they still need to be modeled to accurately represent the effects that they might have on the electric field distribution. Finally, of course, that if you're using tES in patients that have ongoing medical conditions like epilepsy, stroke, or chronic pain patients, there's always a possibility of some side effects, even though none have been reported in tES so far. But these techniques should always be performed in the presence of a clinician. If you're applying this technique to patients that are under some sort of drug medication, it's important to control that. It's important that they do a full disclosure of the drugs that they are using, because some drugs can affect can have an impact on the effect of tES. Another important aspect related to applications of tES is the sham protocols in tES. Usually sham in tES is performed with the FISFO protocols. So a FISFO protocol is a protocol in which you have a fade-in period of 30 seconds at the beginning of stimulation. Then you have a plateau of 15 seconds, and then a fade-out period of 30 seconds. And sometimes this is repeated again at the end of the stimulation. This is an effective way of doing sham in the sense that there's no current getting into the brain. But the problem here is that patients can actually tell the difference between this and real stimulation. So in this study where the authors compared sham and active stimulation in a montage with 2 electrodes, and what happened is that after the study, they asked the patients if they had received sham or real stimulation. And most patients that received sham stimulation replied that they thought they had received sham stimulation, whereas most patients that received real stimulation reported that they thought they had received real stimulation. So is there a way to improve this simple protocol for generating a sham condition? Well, in this paper that we published last year, we describe a method that uses montage optimization to do this. This method is something we call Actisham. And how does this work? So in this study in particular, we considered a montage that was aimed at exciting the left motor cortex. And you can do this, of course, by placing an anode over C3 and a cathode over FP2. This is the traditional montage. A more elegant approach is, of course, to define a target area in the motor cortex and to run montage optimization to determine the position and the currents of the electrodes that will excite that area. And in this case, this is the montage that was obtained. And now in the Actisham approach, we use the same target area, but now instead of asking that the electric field be excitatory here, we asked that the electric field be very small here, almost 0. But at the same time, to prevent the optimizer to simply say, okay, if you want a 0 electric field, I'll just put all the currents in all the electrodes to 0. We demanded that the currents in the electrodes be of at least 1 milliamp, for instance. Okay? And what we see is that the optimizer gave us a solution where it used an interleaved array of anodes and cathodes that does induce a very low electric field as compared to the other montages in the target area that didn't have any effects on motor cortical excitability. And this is achieved through something called current shunting in tES. And current shunting is something that happens naturally in tES, as I mentioned previously. So if you have 2 electrodes, 1 anode and 1 cathode located close by, most of the current that you're injecting won't get into the brain. It will actually be shunted through the scalp from the anode to the cathode. The Actisham montage, what it does is that it positions the anodes and the cathodes so that this shunting through the brain is actually prevalent over the current that goes into the brain. So almost no current goes into the brain, and most of the current is shunted through the electrode. And of course, that with this protocol, the subjects reported that they felt sensory feedback very similar to the one they would get in the bipolar and the multifocal optimized active montage, whereas that wasn't the case in the sham protocol with the bipolar montage. Again, we have a poster where we describe this Actisham approach in more detail. This poster is available if you follow this link. Finally, I would just like to describe the home application possibility of TCS. It might be important to do TES in a home setting because it's an intervention that requires typically long durations, a period of some weeks. It employs portable hardware, so it's easy to use in a home setting. It benefits populations that depend on others or have trouble moving on their own, like elderly, disabled, or disorder of conscious patients. And of course, it has a reduced cost and it's very convenient for caregivers. However, as all home applications, there's always the threat of misuse. So you need to be careful to control the application of this technique in a home setting. And of course, training needs to be given to the caregiver and patient. However, there's more and more interest, as you can see from the list of publications, that's being generated in applying tES in the home setting. So this is something that Neuroelectrics is looking into. And in the future, we'll have another webinar where we will describe our home solution in more detail. So we're almost at the end of this presentation. I would just like to finish by saying that tES is an exciting field of research. That's why I like to work in it. And there's new developments that happen every day. For instance, one important development is in the field of temporal interference tES, which is a new approach that people are using to reach deeper targets. Basically, what this does is that it uses modulated electric field envelopes that have the maxima of modulation happening in deeper regions of the brain to modulate the neurons in those regions. Spinal cord stimulation is another form of transcranial electrical stimulation that's gaining more and more interest these days. Basically, it uses the same type of tools and equipment. But now the objective is, of course, to modulate activity of the neurons in the spinal cord. And this is being investigated in ALS, MS, and spinal cord injury. And of course, you can do the same sort of volume conductor head models to model the electric field distribution in this technique. And in a paper that we, together with some researchers, published last year, we also describe a new possible application for tES. In this application, what we did was that we used montage optimization to determine those parameters to target the glioblastoma in patients with this type of tumor. And we found that tES led to a reduction of tumoral perfusion in these patients, which might have some effect in life expectancy for these patients. This is something that we need to see more in future studies. But this is very important because these patients, unfortunately, they don't have a lot of treatment options. And just conclude, let me just say that we here at Neuroelectrics believe that tES is a very important technique that has the potential to help a lot of people for which treatment options aren't that good today. And that's why we continue to focus on this technology, and we are committed to improve our product line and offer services that allow people to fully use the potential of this technique. I would also like to encourage you to visit our website. It's been newly redesigned. It has this lovely design now. And in that website, you'll find a lot of ebooks and white papers that are a very good starting point for you to learn more about tES, about EEG. These are really well written. We put on a lot of work on writing these resources. They are freely available, so this is a great point for you to start to learn more about this technique. If you liked the part about computational head models and you think that you want to learn more about it, we offer these services also in Neuroelectrics. So please visit our website and ask us for more information about this. And make sure you check out our future webinars. The next webinar is scheduled for the 23rd of April. And it will be given by my colleague Laura Val, and it's going to be about StarSteam and about using StarSteam for research with tES and EEG. So thank you for this time, and now I'm available to answer some questions.